My name is Bill Pronto, and I'm here to start off session number 2, which talks about some of the preclinical animal studies evaluating in utero gene therapy. Thanks for that first great session. Hopefully, we'll continue. And so during this session, each of the speakers is going to highlight some particular diseases for which there are preclinical studies and which we think may hold promise as a future avenue for prenatal gene therapy. I'm going to speak specifically about in utero gene editing for lysosomal storage diseases and touch a little bit on monogenic lung diseases. So what is the problem? This was alluded to earlier during the first session, but the problem essentially is birth defects. They're common, with 1 in 28 babies born with a birth defect, costly and merciless. They're mysterious. They're frequently overlooked with research underfunded, and they're deadly. Birth defects are a leading cause of infant mortality. And within this category of birth defects, the World Health Organization, as well as other organizations, estimate that up to 10,000 human diseases can be attributed to monogenic birth defects, or genetic defects in a single gene. They result in over 40% of hospital admissions, cause significant pediatric morbidity and mortality, and many of them can strike prior to birth, as we just heard. And unfortunately, there are limited treatment options for a lot of these disorders. Now, with the advent of gene editing technologies, including CRISPR-Cas9, among many others, there is now this hope for a potential one-time, single-shot cure for a lot of genetic diseases, including these monogenic diseases. And just to set the table first, when you think about gene editing as it would be applied as a therapeutic approach, there are 2 broad categories, ex vivo and in vivo gene editing. As many I'm sure are aware, ex vivo gene editing involves harvesting of cells from a patient's body, editing them outside of the body, and retransplanting or infusing them back into the patient. In vivo gene editing involves delivering the gene editing technology directly to the patient in some sort of delivery vehicle, whether it be a lipid nanoparticle or a viral vector, as Stefan had pointed out earlier. And just again, to clear the table, when we're thinking about gene editing as it would be applied to a fetus, we're predominantly talking about in vivo gene editing and not the removal of fetal cells and modifying them ex vivo. Now, gene editing traditionally begins with instituting a double-stranded DNA break, and this is a slide courtesy of Fyodor. And after that double-stranded DNA break is made, the cell repair mechanisms then take hold, and one of 2 things can happen: either non-homologous end joining, in which the break is repaired and bases are either inserted or deleted, ultimately resulting in the silencing of a gene, or it can be used to cut out a small segment of DNA. Alternatively, if a repair template is provided, when the double-stranded DNA break is repaired, the DNA sequence that's provided in the repair template is incorporated into the DNA, and this could in theory be used to fix an underlying pathogenic mutation. The great news is, as alluded to earlier, there are a number of ongoing clinical trials evaluating in vivo as well as ex vivo gene editing for a number of diseases after birth. Here are some that are listed. Although the traditional gene editing with CRISPR-Cas9 and other modalities has involved using double-stranded DNA breaks, there have been evolution of gene editing technologies in which double-stranded DNA breaks are not required. One example of this is base editing, developed by David Liu. Base editing does not require making a double-stranded DNA break. It can fix specific bases based on whether it's a purine or a pyrimidine, changing it to the other purine or pyrimidine. And it's, in theory, it can be more efficient than traditional CRISPR-Cas9 gene editing, such as homology-directed repair. Now, with this brief background on gene editing, I'd like just to highlight again what Tippy had mentioned, that we're now at a lucky time in medical history where the advent of prenatal diagnostics make it such that most genetic diseases can, or sometime in the future, will be able to be diagnosed before birth. Some of these diseases cause significant morbidity and mortality before birth. And so the ability to diagnose these diseases before birth, combined with their morbidity and mortality, begs the question of instituting a fetal therapy, such as prenatal gene editing. This is the rationale for in utero gene editing, again highlighted by Tiffany during the introductory talk. I guess I would just like to highlight 2 things as it pertains to gene editing. For gene editing, since we're talking about changing the endogenous sequence of the DNA to a corrected version or a therapeutic version, it'd be ideal to do that in a stem or progenitor cell population. Therefore, any changes that are made could be passed on to subsequent cell populations and daughter cells. It's important to note that in the fetus, in addition to the hematopoietic stem cell, which was mentioned earlier, stem and progenitor cells of a number of other organs are potentially more accessible and easy to target. Another thing I'd like to highlight is that the ultimate goal of any of these therapies that are applied before birth is to institute a cure before the onset of irreversible pathology. This again speaks to identifying diseases for which the pathology begins before birth and carefully weighing the risk-benefits of treating something before birth. When we think about potential diseases that would be amenable to prenatal gene therapy, including gene editing, one area of interest that we hold is in using gene editing to treat lysosomal storage diseases. So lysosomal storage diseases consist of a number of different diseases. They're inherited metabolic disorders characterized by abnormal buildup of toxic materials in lysosomes. Depending on the disease, they're due to a deficiency in an enzyme, an activator protein, or a transporter. These diseases can affect multiple organs, including the CNS, skin, heart, brain, or musculoskeleton. And for many of these lysosomal storage diseases, as demonstrated by the table here, the pathology begins before birth. In the upper right here, you see an image of a newborn baby prenatally diagnosed, that was diagnosed with MPS 7 or SLY syndrome. We heard the very nice introduction from the parent of a patient with SLY syndrome. And as you can see, shortly after birth, you can identify sequelae of disease. The patient has bilateral inguinal hernias, ascites, abdominal muscle weakness. This obviously all began before the baby was born. In the bottom picture here, we have an EM of the hepatocyte from an 18-week gestation fetus that was diagnosed with Hurler syndrome or MPS 1H. The pathognomonic signals of this lysosomal storage disease can be seen in this EM picture here. Again, just highlighting that for some diseases within the lysosomal storage disease category, the pathology is known to begin before birth. In our lab, we have an interest in looking at prenatal therapies for MPS 1H or Hurler syndrome, the disease for which the EM I just showed you was discussing. MPS 1 has a prevalence of 1 in 100,000. It results from mutations in the IDUA gene, resulting in a deficiency in the IDUA enzyme. And there are a number of different mutations that cause this disease. However, there is one predominant mutation, a G to A mutation in this gene, which is responsible for a severe form of the disease. Again, speaking to the importance of a genotype-phenotype correlation when you're thinking about prenatal therapies, as Stefan had mentioned. As I just mentioned, the pathology for Hurler syndrome is believed to begin before birth. And unfortunately, there are limited treatment options available for patients born with this disease. Currently, treatment options include enzyme replacement therapy, as well as hematopoietic stem cell transplantation. These treatments, although game-changing for some patients, are associated with significant morbidity and mortality. There's a limitation of finding matching stem cell donors. There's also issues with compliance for enzyme replacement therapy. And finally, even if everything goes perfect and the treatment is initiated, it is known that these two therapies do not treat some of the underlying musculoskeletal and cardiac phenotype associated with this disease. And so when patients are born with Hurler syndrome, they demonstrate signs and symptoms of the disease shortly after birth. And as they grow older, they have a worsening progression of the disease. Unfortunately, by 10 to 15 years of age, most patients with Hurler syndrome can die from cardiorespiratory failure. So we think that this would be an ideal disease to study the feasibility and the potential of in utero gene therapy and gene editing. The great news is there is already a postnatal clinical trial for in vivo gene editing in Hurler syndrome and is run out of Sangamo Therapeutics. This clinical trial was based upon, among other things, data from mouse studies. And the catch of the clinical trial is that they used zinc finger mediated gene editing technology to introduce the wild type IDUA gene within the albumin locus. Within the albumin locus, with the idea of using the albumin promoters to express the IDUA gene at therapeutic levels. This is an ongoing clinical trial and be excited to see how this turns out. We looked at in utero gene editing in a mouse model for Ehlers syndrome. Specifically, we used an AAV vector to deliver an adenine-based editor in the mouse model. Fetuses were born and then followed for long term, up to 6 months of age, at which multiple outcome measures were assessed. What we were able to find is that we had significant editing in the heart and the liver, with low-level editing in other tissues, including the brain. This editing translated into improved IDOA activity in the serum, as well as in other organs, specifically the heart and the liver. And then the question was, does this have a phenotypic effect? So it's great to have a change in the DNA or the enzyme, but we want to make sure we have a phenotypic effect. So we studied the musculoskeletal phenotype in these mice using CT scans, and we were able to show that in utero treated mice demonstrated improvement in certain musculoskeletal phenotypes. And then importantly, we also looked at the cardiac phenotype. If you remember, a lot of these patients will unfortunately die from cardiorespiratory failure of the disease. When we look at the cardiac phenotype in these mice, we again saw that the prenatally edited mice demonstrated improvement in some of the cardiac phenotype parameters. Histologically, we saw decreased alcine blue, which is associated with disease improvement in mice that were edited as fetuses. And this all translated into an improved survival of prenatally edited mice that was equivalent to wild-type B6 mice and much better than the 50% survival of diseased mice that were untreated at 6 months of age. So this gave us hope, and it was encouraging. We were also interested in assessing whether or not this gene editing therapy could be applied postnatally. And here you see prenatal versus postnatal editing, with the postnatal editing demonstrated by the yellow marks. And we were happy to see that we could achieve relatively good postnatal editing in the heart as well as other organs. This editing was slightly lower than prenatal editing. And then when we looked at the phenotypic effect of this editing, we saw improvement in the phenotype of postnatally edited mice that was not as good as those that were edited prenatally, but it was also was good and provided hope for this potential therapy in the future. The other potential benefit of prenatal gene therapy and gene editing is avoiding a limiting immune response. And there have been a number of studies, including those out of Matt Porteus's lab, which have demonstrated the pre-existing adaptive immunity to Cas9 proteins in humans, likely related to natural infection by strep and staph. Other studies in the mouse model have shown that this pre-existing immunity can limit gene editing in the liver. And so we too looked in our mouse models to see if we elicited an immune response to Cas9 following prenatal versus postnatal editing. And as you may guess, we were able to avoid an immune response following prenatal editing. However, we did elicit an immune response to Cas9, which serves as the backbone for the base editor following postnatal editing. And now I'm going to briefly move on to monogenic lung disease. We think this is a disease category which also holds potential benefit for treatment using in utero gene therapy or gene editing techniques. So congenital monogenic lung diseases, including alpha-1 antitrypsin deficiency, cystic fibrosis, and surfactant protein disorders, can lead to respiratory failure and death shortly after birth with chronic lung disease. Current treatments predominantly focus on symptomatic relief and or culminate in lung transplant. And as you can see by this table here, there are a number of patients with CF and surfactant protein disorders that die awaiting a lung transplant. And even if a lung transplant is successful, there is limited 5-year post-transplant survival. So there's a critical need to develop new therapies for these disorders. Importantly, the CF, surfactant protein disorders, and alpha-1 antitrypsin all have predominant disease-causing mutations that are associated with a severe form of this disease. I'm going to focus on surfactant protein disorders, as Dave Stittleman, who is following me, is going to talk more about CF. Surfactant protein disorders can be grouped into 3 main types. And surfactant protein B and ABCA3 disease is associated with respiratory failure shortly after birth, with patients dying in the neonatal period or early childhood if they don't receive a lung transplant. We sought to study the feasibility of in utero lung gene editing in 2 different models. One is a fluorescent reporter model shown here on the left, and the other is that of a surfactant protein C deficient model. In the fluorescent reporter model, we were able to show that we could target pulmonary epithelial cells following in utero delivery of Cas9. This is critical because these are some of the cell types that you'd want to target for many monogenic lung diseases. And then the surfactant protein deficient model is a gain of function model in which accumulation of the misfolded protein is toxic to the lungs. Over here, you can see a homozygote mice where the lung essentially looks like liver, and you can understand why these mice die within a couple hours of birth. In utero gene editing to remove the gain of function mutated gene resulted in improved histology in these mice and improved survival. Most mice that were not treated died by 6 hours. We were able to achieve a 6% survival, which isn't great, but it's a good starting point, and mice that underwent editing. And then finally, I'll just touch on the specific safety concerns of in utero gene editing. As Tippy alluded to, there are 2 patients that are involved, the mom and the fetus. And when I think about safety and risk, I put it in 2 different baskets: procedural risks and risk of therapy. We're going to talk more about the procedural risks later on in the day. Therapeutic risks involve the risk of the delivery vehicle, as well as risks of the editing technology that we're delivering. From a editing technology perspective, we have looked at the risk of maternal editing as well as germline editing in these mouse models. We were able to show that there was no significant evidence of maternal editing in our mouse models, nor do we see any evidence of germline editing in the mice that were edited as fetuses. We did this in 2 ways: by looking at their gametes as well as by mating edited mice. And so that's it. I hope I've been able to convey to you that in utero gene editing with CRISPR technology is feasible in the mouse model and can rescue clinically relevant human disease models. Delivery platforms, including AAV and nanoparticle technology, hold promise and need to be evaluated. However, I think it's important to stress that there are ongoing and recent safety concerns regarding AAV-mediated postnatal gene therapy, and these concerns highlight the critical need for future small and large animal studies, potentially non-human primates, to evaluate the safety and efficacy of in utero therapy using these delivery approaches. So I'd like to thank you, and I'm happy to turn it over to David Stillman from Yale University.
Thank you, Bill. So I'm a pediatric surgeon at Yale New Haven Children's Hospital, and I run a translational laboratory focused on fetal therapy. This is my disclosure. So I'm going to talk today about particle-based fetal gene editing for hemoglobinopathies like sickle cell anemia and specifically thalassemia and cystic fibrosis, which are common lethal genetic diseases. My laboratory has expertise and interest in fetal therapy, and this work was made possible by experts in molecular therapy and biomedical engineering at Yale. This is our team of scientists, so senior scientists in biomedical engineering, molecular therapy, and in our Cystic Fibrosis Center, but also graduate students and surgical residents who are sort of the lifeblood of the laboratory. So the advantages of fetal therapy have been well covered in this meeting so far. A feature that I wanted to point out in this talk is the unique anatomic and developmental features of the fetus. So this is a graph of hematopoiesis during pregnancy. At the time we would offer intervention, the hematopoietic stem cell, or HSC, is in the fetal liver, and those migrate to their final location in bone marrow later in development. It should be pointed out that the fetus is developing an immune system. It does not have a full complement of T cells and B cells and is hypoimmune compared to postnatal life. And that's important in terms of potential reaction to foreign vectors or gene products. For the experiments I'm going to show you, we use editing reagents that are a little bit different than CRISPR or zinc finger nucleases. We use something called peptide nucleic acids, or PNAs. So a PNA, instead of the sugar phosphate backbone of DNA, has a protein-like backbone and can be built and designed with the same bases as DNA or RNA. And they can be designed to bind specific sequences of DNA. This cartoon displays how our editing works. So the black bars represent the chromosome. The frowny face is the mutation in that chromosome. A PNA is designed to bind upstream or downstream to the mutation in the chromosome. The cell's editing mechanisms find the bound PNA and excise that segment of DNA. We also deliver a homologous segment of DNA that's modified to correct the mutation. This donor DNA is then used as a template for homologous recombination. It's very important to note that this method uses something called homology-directed repair, or HDR, and nucleotide excision and repair, NER pathways that are very high fidelity. My analogy for how these editing reagents work is that the chromosome is like a Microsoft Word document. The PNA is like the find function, and the DNA is like the replace function. And it's important to note that gene editing has an advantage over gene replacement therapy in that the regulation of the gene remains the same and that we're not limited by the cassette size of viral vectors. And then finally, to facilitate entry of these agents into cells, they're loaded into biodegradable and biocompatible nanoparticles. The experiments I'm going to show use unmodified particles made from a polymer called PLGA, but they can be made from different polymers. The surfaces can be modified with different chemicals, and the surface can be modified with cell-specific ligands or antibodies. Sorry. The PNA-DNA editing reagents are effective in hematopoietic stem cells ex vivo in mouse and human cells. As has been mentioned, ex vivo editing has been successful in hematopoietic stem cells ex vivo using CRISPR. The Glaser lab has made several modifications to these PNAs to improve binding, and they've been found to be most effective in stem cells. So this is an RNA sequencing experiment looking at the pathways that mediate the editing in fetal above and adult below hematopoietic stem cells. These pathways appear to be upregulated in fetal cells, making the fetus an ideal patient for this strategy. So what I picture clinically is that under ultrasound guidance, fetal blood vessels or the amniotic cavity will be accessed by a needle. Fetal blood transfusion, where the umbilical vein is cannulated and the fetus receives a blood transfusion, has been performed since the 1980s and carries a very low risk of fetal loss. So from a technical standpoint, fetal gene therapy can be delivered in a safe and minimally invasive fashion. This is a video, hope it works, of an intravascular injection in a mouse. We cannulate the vitelline vein and inject, in this case, fluorescent nanoparticles, and this joins the hepatic circulation of the fetus. IV injection of green fluorescent nanoparticles results in remarkable uptake in fetal liver. You can see it's distributed actually throughout the fetus. Again, at this time point, the fetal liver contains the hematopoietic stem cell, which will then migrate to the bone marrow to become the adult hematopoietic stem cell, which is vital for lifelong correction of hemoglobinopathy. In hundreds of injections, we've seen long-term survival, normal growth, and no changes in inflammatory markers after delivery of these agents. So our disease model, one of the diseases I want to talk about today is hemoglobinopathy. So sickle cell anemia and beta thalassemia are the most common lethal genetic diseases globally. A single systemic dose of gene editing before birth could cure these diseases. In vivo and systemic editing of HSCs could democratize therapy for sickle cell and thalassemia across the world. Our mouse model has beta thalassemia, which is a severe genetic disease caused by mutations in the beta globin gene that significantly reduce adult hemoglobin, resulting in chronic anemia, lifelong dependence on blood transfusion, and in order to survive, patients require blood transfusions every 3 to 4 weeks to maintain adequate hemoglobin levels. Transfusions temporarily improve the anemia. They don't address the underlying genetic cause. Transfusions also lead to iron overload, which can result in progressive multi-organ damage and failure. For lifelong treatment of these blood diseases, correction of HSCs is a viable strategy. In September of 2021, Bluebird Bio submitted a biologics license application, or BLA, to the FDA for lentiviral-mediated gene replacement therapy in adults, adolescents, and pediatric patients with beta thalassemia. This involved ex vivo correction that involves harvest of the patient's hematopoietic stem cells, treatment with viral vectors, ablation of the host bone marrow, and administration of treated HSCs. I hope to demonstrate a related but different approach. So systemic delivery of editing reagents in the fetus obviates the need for cell harvest, manipulation, and host ablation. So for these experiments, we use a mouse model of beta thalassemia and gave systemic doses of nanoparticles loaded with editing reagents to treat this mouse model. Those particles go to the fetal liver, which again is the site of the fetal hematopoietic stem cell. And again, this was done without any ablation of the host. We demonstrated 6% editing of bone marrow without detectable off-target effects at homologous sites. So these were fetal cells that were corrected in their native niche before birth. This results in correction of anemia. So these mice in the center genotype as having beta thalassemia, but they're not anemic like their untreated counterparts. Reticulocyte count is a marker of the need to make new blood cells in anemia and is normalized in treated animals. And finally, there is a survival advantage in the long term over the course of over a year. We also saw normalization of the peripheral smear of the blood. Anemic animals make blood in their spleens, and their spleens become gigantic. Treated animals demonstrate a more normal splenic size and more normal splenic architecture. And so the next step in this project, in my mind, is testing different nanoparticles. So this was really our first stab at this approach. Nanoparticles can be made from different polymers and have different surfaces and different loading characteristics, and they can be made with different sizes. So we've tested diluted particles after systemic fetal delivery and done flow cytometry for fetal hematopoietic stem cells. This arrow points to the number of HSCs that had uptake of particles. It appears that as we change the size and type of particle, we can drastically improve the number of treated hematopoietic stem cells. I now want to turn our attention to cystic fibrosis. So there are several mutations that cause cystic fibrosis, but the most common is a 3-base deletion that results in a loss of phenylalanine at position 508 of a protein called the cystic fibrosis transmembrane regulator, or CFTR. CFTR is a transmembrane ion channel that sits on the surface of several vital epithelia. So normally chloride passes through this channel, sodium and water follow, and that results in perfect mucus. If CFTR is impaired, chloride does not flux, sodium and water do not follow, and the mucus becomes thick. Thick mucus in the lungs drives most of the mortality in cystic fibrosis. The duct to the pancreas and biliary tree become obstructed with mucus, and over 80% of babies with cystic fibrosis have a burnt out pancreas at birth. And we, as surgeons, see babies with impacted meconium who require bowel surgery for bowel obstruction in the first day of life. So CF is a relatively common disorder where there's organ damage before birth, providing compelling rationale for fetal therapy. And for blood diseases, ex vivo therapy is possible because the stem cells may home to their niche. There may be issues with how competitive the fetal HSC compartment is. But cellular therapy for a disease that impacts multiple epithelial cells in different organs is likely better treated with systemic therapy, where you would treat the cells where they lie. And so we've demonstrated nanoparticle uptake in lung after systemic delivery. And using our first generation nanoparticles, we see about 5% editing in those tissues. We need to do a better job of characterizing what cell types are being edited. We also see uptake in the fetal gut and fetal pancreas, which are vital for treatment. Our group has optimized editing reagents. So this is normal CFTR, the CFTR with the 3-base deletion. We've created PNA editing reagents that recruit innate editing enzymes. And if we give donor DNA that's homologous but altered, we then correct CFTR. As Bill was saying, phenotypic results are the most important. So one of the assays that we do is for nasal potential difference. So nasal potential difference, we measure chloride flux across the nasal epithelium in adult mice. So untreated CF mice cannot generate a negative potential because they don't have chloride flux, but wild type mice can. These mice in the center genotype as having cystic fibrosis, but after systemic fetal editing are able to generate chloride flux. Lung infection and pneumonia is a driver of morbidity in these patients. And we quantified inflammatory cells in the airways of adult mice. CF mice have a lot of inflammatory cells, but treated CF mice look remarkably like their like wild type mice. The next step in this project is similar to the HSC project. So we demonstrated that nanoparticles made from different polymers have higher uptake in epithelial cells than particles in our initial experiments. And so we need to test the efficacy of editing using these improved polymers. We also need to establish the safe range in larger animal models. And then in conclusion, clearly we're early in the process of studying particle-based fetal gene editing, but I remain optimistic about these results in preclinical mouse models. I hope that I've demonstrated that unique features, namely stem cell accessibility, make the fetus an ideal candidate for this line of therapy. And with continued optimization, I hope someday that one dose of systemic therapy could cure otherwise devastating diseases before birth. So now I'd like to introduce Graça Almeida Parada from Wake Forest. Thank you for your attention.
Thank you, David, and thank you, the organizers, for giving me the opportunity to speak. So the hemophilias are X-linked disorders caused by mutations in human coagulation factor 8 or factor 9 genes. These mutations lead to missing or defective factor 8 or factor 9 proteins, which are essential blood clotting proteins. Persons with severe hemophilia, we have less than 1% of these factors in circulation, suffer from severe spontaneous bleeding, while patients with more than 5% experience bleeding only after serious injury, trauma, or surgery. Therefore, a small increase in factor 8 or factor 9 has a substantial effect on patients' quality of life, making these diseases ideal targets for gene therapy. Importantly, 71 to 78% of persons with hemophilia have a family history of the disease, allowing for prenatal diagnosis and intervention. Although severe hemophilia A and B are clinically similar, there's differences in incidence, size of the affected gene, type of mutations, and the risk of inhibitor formation. And these may dictate different approaches to treat these two forms of disease. So given its small size, for factor 9, both gene addition and gene editing would be feasible. However, for hemophilia A, in addition to the large size of factor 8 gene and factor 8 protein, the choice of the delivery method will also have to be taken in consideration. But overall, both are very attractive targets for gene addition. There are a vast array of products available to treat these patients, and the standard of care in countries that can afford it is prophylactic replacement protein. Recently, new products that allow subcutaneous injection have either come to the market or are available as investigational drugs. But although these therapies have advanced the standard of care and the burden of disease in persons with hemophilia, this burden of disease continues to be high. A meta-analytic study for Data and Demographics Committee of the World Federation of Hemophilia demonstrated that for those born with hemophilia, the chances of living a life of normal duration and quality will be reduced by 33% in high-income countries and will be further reduced to 93% in low-income countries. This is because 80% of the patients around the world do not have access to treatment. In those with access to treatment, the current therapies do not completely prevent bleeds. And importantly, 25 to 40% of hemophilia patients have a lifetime risk of developing inhibitor antibodies, increasing the risk of morbidity and mortality in this group of patients. Even with the most advanced treatments, joint disease, development of inhibitory antibodies, and prevention of life-threatening bleeding continues to be a challenge. The promise of an optimal treatment or maybe a cure for the hemophilias arose when new clinical trials using direct injection of different serotypes of AAV vectors and optimized factor 8 here or factor 9 in the next slide transgenes showed therapeutic promise in adult hemophiliac patients. In addition, there are several ongoing or upcoming clinical trials using CD34 cells or MSC transduced with lentiviral vectors to express optimized factor 8 or factor 9 transgenes. Of note, in hemophilia B is the upcoming clinical trial based on groundbreaking by Dr. Kati Haya and colleagues using AAV to mediate zinc finger target insertion of factor IX into the first intron of the albumin locus. Overall, the results from early phase AAV clinical trials demonstrated the safety of gene delivery and showed marked efficacy as patients who received gene therapy had 90% reduction in bleeds and in factor use. However, while patients receiving AAV expressing factor IX maintained 5% activity up to 10 years, in patients receiving AAV therapy for factor VIII, the therapeutic levels dropped significantly at about 5 years, demonstrating there are still unmet clinical needs for these patients. The drop in efficacy in AAV clinical trials for hemophilia A could be due to the inflammatory responses seen in these patients that received the therapy. Of concern is that administration of a second dose of the same therapy would be challenging due to the immunization against the capsid. Even the first dose can be only administered in pre-selected patients, since pre-existing immunity to AAV can be as high as 60%, depending on the age of the patient and the AAV serotype, impacting the ability of patients to receive the therapy. Of note is that newborns already have antibodies against certain AAV serotypes. In addition, the episomal nature of the vector makes the AAV application to pediatric patients challenging and may lead to the dilution of loss of the vector as the liver grows. Of concern is that data in dogs and mice indicate that AAV can also integrate, and the mice data in mice also suggests that active cell cycling may have potential to trigger tumorigenesis after AAV delivery. Thus, preclinical studies modeling pediatric populations are urgently needed, since data from adults cannot, in this case, be extrapolated to the age group. Indeed, there's an urgent medical need to specifically develop curative treatments for neonates and infants with hemophilia and develop appropriate and safe clinical trials for this age group. Bleeding patterns in newborns differ from children that are older and in adults. These patients, neonates bleed 44% of patients with hemophilia A had a bleed by the 1 month of age. 10% received replacement therapy within the first 24 hours of life. These patients have increased risk of cranial bleeds, and because the treatment is on demand and when needed, the frequent administration of the therapy places an extreme treatment burden in patients that must receive these therapies through a central line. Since inhibitors developed during the initial 20 exposure days, alternative treatment approaches that could avoid inhibitor development would change the paradigm of hemophilia A treatment. So, which kind of therapeutic approaches can we offer these patients that would provide the next great advance in care? We think that hemophilia A is an ideal disease to treat prior to birth. 75% of patients have a family history. Prenatal diagnosis is available and used to either optimize obstetrical care or any other options. Prenatal treatment could preclude the development of inhibitory antibodies to the missing protein. The prenatal gene therapy could avoid immune response to the transgene, and a small amount of protein in circulation has high therapeutic benefit, as 5% would turn a severe to a mild disease. And patients could be cured with a single treatment. There are many studies led by us and others demonstrating the promise of in utero gene therapy using viral vectors encoding a variety of transgenes to induce postnatal tolerance to the encoded protein. Initial studies using in utero gene therapy using adenoviral vectors to deliver factor IX made the important observation that in utero delivery induced immunotolerance to factor IX, allowing immune challenging postnatally without inducing inhibitors. A similar study using a more clinical relevant vector, AAV, Sabatino and Mackenzie show that in utero injection, followed by postnatal readministration of adeno-associated virus, AAV1 in this case, resulted in long-term expression of factor IX. Importantly, cellular tests of immunity to challenge the mice that received the therapy demonstrated that the mice did not have a cellular or humoral immune response to factor IX, whereas all mice injected at the adult stage developed antibodies to factor IX. Also using AAV, but performing in utero transplantation in fetal sheep, David et al. Demonstrated that factor IX was present at earlier times point post-therapy, but these levels dropped as the fetal liver and lamb weights increased. No functional antibodies to factor IX protein or AAV vector were detectable, but by contrast to the murine studies, lambs mounted an antibody response after injection of factor IX protein with Freud's adjuvant. Recent studies by Matter et al. Reporting on a long-term follow-up of 4 out of 6 animals, monkeys, treated with AAV carrying a transgene for factor IX, and these studies were performed in late gestation, and as I said, more than 6 years post-treatment, demonstrated that these animals continue to express factor IX at therapeutic levels, but the long-term expression actually was higher in males. Low frequency integration was found with random genome-wide hepatic proviral integrations, but no integrations in hotspot sites. Another study using monkeys, fetal monkeys, this time performing in utero gene therapy at early gestation at 0.4 times of gestation. Animals received either AAV5 or AAV8 at doses at 4 times to the 12 to 4 times 10 to the 13 viral genomes per kilo per birth weight, and they were monitored throughout and after infancy. 2 out of the 3 animals that receive AAV5 have subtherapeutic levels of factor IX. By contrast, AAV8 recipients had sustained factor IX expression despite the rapid growth, and fared similarly with median factor IX expression in the therapeutic range. This study demonstrated that in utero gene therapy did not sensitize the animals to AAV capsid, and animals did not develop antibodies against factor IX. In addition, maternal AAV responses and anti-factor IX responses were not detectable in a mother. So, although these studies using direct gene delivery of vectors are very promising, and I'm sure they'll go forward. Also, there's another type of gene therapy that implies the use of cells as vehicles for gene delivery. And this enables multiple safeguards as copy number and integration site analysis can be implemented in production. This approach would also eliminate the possibility of off-target effects, making a cell-based approach far safer in a prenatal setting. So we and others have shown the ability of deliver factor VIII transgenes through cells. And here we used fetal sheep as a model at an age corresponding to that of 18 to 20 weeks in humans, with human placental cells being transduced with codon optimized engineered factor VIII, here called M283, at a dose of 10 to the 7 to 10 to the 8 cells per kilo, and the cells were producing 5 to 10 international units per 24 hours per million cells. Of these 23 animals that were transplanted, 14 were available for long-term evaluation, and 9 were either euthanized or lost to natural causes unrelated to procedure or the therapy. The animals were born, and we followed these animals for at least 3 years after in utero transplant. And evaluation of factor 8 by APTT, you can see in this slide, were expressed as an increase over control, demonstrated that these animals had an increase in plasma levels of factor 8 activity that reach an average of 40% above normal. The red dotted line here represents 20% increase over control, and the solid lines show the mean in factor 8 activity for each one of these animals. More importantly is that the levels of factor 8 activity were maintained throughout these 3 years, despite the exponential growth of the animals that at transplant weighed around 100 grams, and by 1 year of age, they weighed 50 kilos, and now they weigh between 70 and 100 kilos. We investigated whether this therapy caused liver toxicity. We test liver enzymes, and they remain normal throughout these 3 years. We also looked at hematological alterations, and the animals maintain normal white blood cell and red blood cell counts. Throughout these 3 years, none of the animals mounted an immune response to the product, as demonstrated by the lack of IgM or IgG against factor 8. Also, we looked at factor 8 reactive memory, Th1 or Th2 cells, and performed ELISpots, and none of the animals had factor 8 specific Th1 and Th2 cells, while here with these black triangles, you see that the cells were still able to proliferate when they were stimulated with PHA. We also looked to see if the animals that received treatment prior to birth had HLA-specific antibodies directed towards the product, and we used a clinical Luminex assay optimized for sheep, and none of the animals transplanted had a PRA positive score. Lastly, histopathological examination showed no evidence of any lentiviral vector-related or procedural toxicity in any tissue examined, and there were no foci of hyperplasia or neoplasia in the tissues examined. In conclusion, in utero gene therapy can change the paradigm for how we treat the hemophilias. In utero gene therapy promises the birth of an healthy infant, or at least provides a viable treatment to an at-risk population for whom not many therapeutic therapies are currently available. Data in mice, sheep, and monkeys demonstrate that early in utero gene therapy could preclude the development of antibodies to the missing factor 8 or factor 9 protein without sensitizing to the capsid. In our studies, we show that the placental cells, when transduced with a codon-optimized engineered factor 8, resulted in curative plasma levels of factor 8 following birth and product-specific immune tolerance in the preclinical large animal model for at least 3 years post-birth. Treatments in the fetus, newborns, and young children may require specific preclinical studies due to the marked differences in fetal anatomy and physiology compared to the adults. And I would just like to thank the colleagues that I present their work in my presentation, my group, and the students that worked on these studies, and NIH that supported the studies. Thank you. And now I would like to introduce Christopher Parada from Wake Forest Institute for Regenerative Medicine.
Good morning. I'd like to start by thanking the organizers for the opportunity to speak with you today about work that's been published to date on the risks to the fetal germline as a result of in utero gene therapy, which I will hereafter refer to as IUGT. So in one of the earliest studies exploring delivery of an exogenous gene to the hematopoietic cells, by directly injecting an MMLV-based vector into fetal sheep, we observed that all tissues analyzed harbored the provirus, including the reproductive tissues, thus raising the possibility that this approach to IUGT might actually place the germline at risk. This led us to ask how we might be able to prevent these undesired off-target effects and thereby increase the safety of this approach to IUGT. In some of our first studies, we found that by simply altering the time during gestation that the vector is administered, makes it possible to dramatically affect which tissues are transduced and the level of transduction within those tissues. We then showed that these changes in tissue tropism as a function of gestational age were the result of developmental changes in the levels of the amphitropic receptor, or PIT2, that was expressed on these tissues during gestation. These findings raise the exciting possibility that it may be possible to target desired tissues or cell types by simply pseudotyping the vector with an envelope whose receptor is expressed on the desired tissue or cell type and is absent from non-target tissues like the germline. Now, going back to the germline specifically, is the germline itself actually at risk following IUGT? Well, our PCR data suggested that may be the case, but we had simply looked at unfractionated reproductive tissue, so this wasn't really conclusive. In some of the first studies to explore this issue directly, Rudolf Janisch injected wild-type MMLV into mouse embryos and showed not only did the provirus integrate into the germline, but the provirus was then passed to subsequent generations in a Mendelian fashion as part of the permanent genome. But Janisch's studies used wild-type virus and were performed during early embryonic development, so they likely grossly overestimated the true risk of germline modification. Nevertheless, myriad studies have now shown that germ cells in large animals are susceptible to transduction with a variety of viral vectors, and that this technology can actually be used to fairly efficiently generate transgenic animals. So to rigorously address whether the germline is in fact at risk as a result of IUGT and determine the frequency at which this might occur, we used a multi-pronged approach to conduct studies over a period of more than 8 years using the sheep model. We performed breeding studies, albeit fairly limited due to the large size of the sheep and also the long gestational period of 5 months. We performed PCR on ovipure isolated sperm cells. We then further purified the sperm using a forensic purification method and performed PCR, and ultimately used FACS sorting to isolate pure sperm cells and perform PCR. We also performed immunohistochemistry, or IHC, on sections of the reproductive tissues of these animals. These studies led us to the following conclusions. So in our breeding studies, we saw no proviral DNA in the offspring. We analyzed a total of about 3 billion purified sperm cells by PCR. We analyzed over 65,000 germ cells in tissue sections. From these analyses, we found that only males exhibited germline alteration. Only 6 of the 19 IUGT sheep were PCR positive in their sperm for provirus. And of these 6, only 2 exhibited transgene positive germ cells in the testes sections. In animals with the altered germ cells, the incidence of germline modification was roughly 1 in 6,250. Now, we found this number to be interesting because a publication by Haig-Kazazian in Nature Genetics had quoted, and I'm not exactly sure where this number came from, that the regulatory limit for germline alteration was about 1 in 6,000. So the incidence of germline modification we saw was actually below this limit. Importantly, 1 in 6,250 was orders of magnitude below the rate of endogenous retrotransposition mediated insertional mutations in the male germline, which has been reported to occur in about 1 in 50 individuals. Now, this led us to ask the question of what factors determine the risk to the fetal germline. So to address this, we performed detailed IHC studies on the testes of fetal sheep at varying stages of gestation using markers that are known to be expressed on human germ cells at various stages of differentiation. We found that germ cells expressing SSEA1 are migratory and are uncompartmentalized until about 70 days of gestation. The germ cells then began to express SSEA3 as they commenced the process of compartmentalization within the forming sex cords. Upon compartmentalization, the germ cells began uniformly expressing SSEA4, and they continued to do so throughout the remainder of gestation. Now, in our studies, we employed MMLV vectors, which require cell division in order for genomic integration to occur. As you can see in this slide, the germ cells don't actually begin undergoing significant cell cycling until they have become compartmentalized within the sex cords, and they would thus be presumed to be protected from viral vectors by the blood-testis barrier. Now, these studies, to kind of summarize, revealed several aspects of male germ cell behavior, which we think may likely impact the risk of inadvertent modification following IUGT. First, most male germ cells are not, in fact, compartmentalized during early to mid gestation, and they thus may be unprotected or at risk of modification. Male germ cells appear to be largely quiescent until they are compartmentalized, so vectors requiring division will not transduce them very efficiently. Sertoli cells that form the barrier of the sex cords will likely take the majority of the vector hit. And our data suggests that germ cells will be most vulnerable to viral vectors and also to environmental mutagens during passive compartmentalization as cycling begins prior to complete closure of the sex cords. Now, what about vectors that don't require cell division for integration? In studies performed during the same time, we were studying germline risk in sheep using MMLV to achieve IUGT. Dr. Alice Terenthall's group conducted studies in NHPs using lentiviral vectors. In contrast to what we observed with MMLV in sheep, Dr. Terenthall observed no modification to the male germline. Instead, she saw a significant transduction of female germ cells, which was later confirmed by laser dissection of individual germ cells, followed by vector-specific PCR. Sorry about that, my computer's jumping slides here. Now, interestingly, despite the importance of this issue, not a lot of studies have been done since these initial ones in the realm of looking at what the risk to the germline may be as a result of IUGT. I'd like to highlight 3 more recent studies by Jerry Chan's group in which they use AAV vectors to perform IUGT in non-human primates. All 3 of these studies showed the presence of vector and transgene product within the reproductive tissues, often at fairly high copy number. But interestingly, no in-depth studies were done to ascertain whether the germline itself had actually been modified, and if so, at what frequency. So to make some conclusions on data to date on the germline, this data suggests that the risk to the fetal germline as a result of direct vector injection IUGT is likely to be very low. However, given the discrepancy seen in different species and with different vectors, this important risk will likely have to be assessed with multiple animal models using the specific vector to be employed prior to implementing this in the clinic. And I would just like to finish by thanking Graça, all of our lab members, both past and present, Dr. Atala and the whole team at WFirm, and our funding sources, NHLBI and NICHD. Thank you very much.
Thank you so much, everyone, for those wonderful presentations. My name is Agnieszka Chekwitz. I'm an assistant professor at Stanford University in our Department of Pediatrics, Division of Hematology, Oncology, Stem Cell Transplant, and Regenerative Medicine. And I'd like to invite all of the prior presenters to come join us for a panel discussion. So those are some really fantastic presentations that really lead us to imagine the different types of treatments that we can be developing or are developing and that may become available in near term. We're hoping to talk about some of the preclinical science that was presented today. And I believe we have one more presenter that will be joining us as well. Is Dr. Lois Fried here as well?
Yes, unfortunately, I don't have video capability at the moment.
Oh, no worries. But thank you so much, Dr. Fried, for joining us. Do you want to just introduce yourself since you were not introduced earlier as part of the presenters?
Yes, I'm Lois Fried. I'm currently the acting supervisory pharmacologist for the divisions of neurology and the director of the Division of Pharm Tox in the Office of Neuroscience.
Wonderful. Well, thank you so much for joining us. And to all of those in the audience, please feel free to ask any questions and just to put them in the Q&A. And then we can go ahead and ask the various different presenters those questions. I guess I can start us off though, and maybe Dr. Freed, since we didn't have a chance to hear from you earlier, I'll start by asking you a question, specifically in regards to ASOs as another gene therapy modality for consideration. There have been quite a few papers and several groups that have been working on prenatal mouse experiments to test the safety of ASOs. And we were wondering your thoughts on some other important preclinical studies for ASOs. Specifically, what types of toxicity studies do you think would be important to see, especially since many of these targets are species specific?
Sure. Let me just say that in FDA, we don't consider ASOs to be, we consider them to be distinct from gene therapy. So they're handled by a different center than the gene therapy modalities. But, and we have almost exclusively our experience is in the treatment, postnatal treatment of these rare diseases. So we don't have, I don't know if I'm going to be able to comment on specific types of studies. I will say, and I very much appreciate the opportunity to participate in this. And I learned a lot from the presentations, and it seems like the kinds of questions that we would ask if someone were to propose such to us is exactly what has been pursued previously. The concern would be target engagement, and assuming that that had been identified and demonstrated in postnatal patients, but also, of course, all the concerns that we have about this type of therapy would be amplified by treatment of the fetus, for example. Target engagement, the extent, the location of target engagement, when during fetal development would that be possible, how to do interspecies comparisons, considering the differences in the course of development and course of the disease, where the target is and how one would access that target by administration into directly into the brain or the ventricle or so forth. So those are the kinds of questions. So the potential to see target engagement and potential efficacy, but also, of course, the safety of the product. For ASOs, some of these, of course, the majority probably have very little target engagement in intact animals. Transgenic animals would be good for efficacy. For safety, they have clear non-hybridization dependent toxicities, primarily depending on the location. Kidney is a specific target organ of concern. Thrombocytopenia is something that we see with these antisense. Of course, there's always a concern about CNS toxicity. And so all of those things would need to be addressed in some way, but how exactly, what kind of studies would be done is something that I just don't have the information to address at this moment.
Thank you for that. It does seem like there are some specific and unique challenges to prenatal therapies, but at the same time, there are many issues that we face that are similar to those that are shared with postnatal therapies, especially regarding the species variability when we're talking about more directed genetic editing approaches. Does anyone on the panel have thoughts on best ways to address that and to be thinking about how do we develop the right models and use the right tissues to be able to show that type of efficacy prior to launching into studies in the target engagement specifically?
I think it's a great question, Nisha. I don't claim to have the answer, but I think you divided it into two separate things, right? The safety of the delivery vehicle and the efficacy of the targeting agent. And if you're talking about editing, then obviously there's a consideration for the DNA sequence, which is going to be different from species to species, right? And so in that case, I think the safety of the delivery vehicle can be assessed in preclinical large animal models like non-human primates rigorously. I think it's, in my humble opinion, I think it's probably difficult to make transgenic large animal models for every disease you want to target. If there happens to be one available, then great. So for Grasa and Chris's work, it's very nice that there is a large animal preclinical sheep model. But in other diseases, that might not be as you might not be as fortunate to have that kind of availability.
I just have one additional comment. Is one of the challenges in thinking about ASO therapies is how to do dose comparisons between the species. And I think that that would be especially challenging when you're dealing with a fetal exposure.
Yeah, there's another question related to that that just popped up in the Q&A chat box. Are there any specific knowledge or differences that should be considered in terms of how fetuses would metabolize or excrete drugs? Do we know enough about that?
I hope you're not asking me, because the answer would be no.
I'm looking at our panel here of fetal therapists that have been doing a lot of these studies, at least in the preclinical setting. It seems like the amounts that we're using are low and very efficacious, but wondering if you have any additional thoughts.
I think based on the work, oh, sorry, go ahead, Russell. Oh, I was going to say, based on the workshop that was just last week, I think, I can't remember when it was, that the FDA participated in as well, discussing fetal pharmacology, pharmacodynamics, and pharmacokinetics. It seems, at least my takeaway from that is that not really a lot is very well understood as far as how the fetus metabolizes drugs. I mean, in this particular setting, I guess we're not discussing administering traditional drugs that would be metabolized, though. I mean, we're administering a viral vector that's going to bind to its receptor and transduce the cell and then be removed. So I'm not sure if the fetal metabolism is a big role. I guess one thing that Grasa mentioned in her presentation was that there are some data in mice, and I think in dogs as well, to suggest that a higher state of proliferation may actually have an impact on genotoxicity as far as viral vector integration. So that's certainly something of concern in the fetus, since basically every tissue is proliferating at the time that we would want to be doing in utero gene therapy.
Yeah, I mean, is what I was going to say. I mean, all of us work on gene therapy or cell and gene therapy. So the metabolizing or the metabolic pathways of drugs in a fetus, it doesn't really apply for in that case. But one thing that I agree with what Chris and Bill said. But one thing that I would like also to bring up to the table is that scale up is very important in terms of gene therapy or cell and gene therapy. And so we have to make sure that the scale up is right and use models that we can at least predict that the scale up to a human is going to be correct. Because so maybe a larger animal model would be more predictive of the dose that are needed to be therapeutic effective in the future. But at the same time, we have also to think about placenta if you want to study the effects of the mother. So I don't think that there's one model that would cover all or one species that would cover all. I think we have to adapt to what are the answers that we want to know. And if it's safety to the mother, maybe we have to do these studies in an animal model that the placentation is very similar to humans to understand the transfer from the fetus to the mother. If we want to just study efficacy, maybe a large animal model, because we are just studying the fetus, we don't care if he's passing to the mother or not, or if we have to consider antibody passage from the mother to the fetus. So all of that, I think each disease is going to be different, and we have to think about all these considerations. What is the question that we want to address? And one model, even for the same disease, we may need more than one model to address all those questions.
Yeah, Grasa, I think that's a great point. And I think it's exemplified even by AAV safety studies postnatally, right? So the concern for integration and oncogenicity has been studied multiple times in mice with AAV, and they show that injecting a neonatal mouse has higher risk of oncogenicity than a postnatal mouse, especially if there's some injury, insult to the liver. But studies in large animal models have yet to bear that out. And so I think you need multiple different models to answer specific questions.
I think that's very important, and that's why it's so helpful to have the models that we currently have for some of these diseases and to be building out additional models. And whereas there are some potential increased safety concerns, there are also the reverse side of that, potential for improved efficacy, both because sometimes that integration might be beneficial for a therapeutic benefit, but also the disease correction earlier in the developmental stage. So how do we weigh those risks and benefits, I think is important to consider, and proud of the work that you guys are doing as you're trying to understand that better. There's another question that was earlier asked about how do we think about when multiple therapies are available for the same disease and not necessarily clinically available, but different approaches could be considered, such as Dr. Grossa, you mentioned for hemophilia. Do we do AAV or do we do ex vivo therapies? How do you think about that, and which paths should we be developing? Should we be developing multiple, or should we be, as a field, picking one? How should we be thinking about that?
I mean, I think in fetal therapies, it should be the same as in adult therapies, right? There's a vast array of therapies that people develop and then see what's the best. So for fetal therapy is the same. I don't think we should exclude any. I think we should bring them all to the table and try to see what is the safest and more and have more efficacy, and that takes time. So I don't think to start with we should just say, well, let's, gene editing, it's good or it's bad. I think since fetus are so different from adults, I don't even think that we can take the safety from an adult to a fetus. So we really have to design these treatments to the population that we are going to apply these therapies. But I don't think we should exclude any ways of treating the fetus. I think we should explore all of them and see what's safer.
To echo what Grassa said, I think each of these technologies work in such a different way, and the technology that I'm using is good for 1 to 3 bases. So it's good for the diseases that I showed, but if you have a disease like alpha thalassemia that's a large deletion, my strategy is ineffective. And questions about do our particles get into brain or not? Right now, the nanoparticles we're using get choroid plexus, but don't get really cortex of brain. So, but if at any point as we continue to study these things, if we see nanoparticles starting to go into brain, then you have a whole new avenue of exploration. So I wouldn't stunt these things in their infancy. I think that you have to continue to test variations. I think that it's going to be, small steps to meet some bar of efficacy and safety. I don't think that we're going to immediately have, a silver bullet to cure a disease. I think that it's, the editing reagents are going to get better. Our ways to get them into cells are going to get better. And I wouldn't, in these phases, discourage someone from testing things.
And I guess the same thing is true for postnatal therapies, very much so as well. This is not a unique conversation to the prenatal therapy space. There's multiple ways of fixing certain diseases, and the field is advancing several of them, and then comparing the safety and efficacies with the hope that each of them provide benefit for patients, which is why we're optimistic about the trials and are launching into them. But at the same time, trying to identify which is the best one ultimately, I think is our goal as a field. And then how do you guys think about finding that optimal dose for these prenatal therapies? Is it similar dose escalation that we do in the postnatal therapy space? Or should we be aiming for that ideal dose initially? I would imagine,
I don't think there's an absolute correct answer. I think for the preclinical studies, you start with the dose that's most effective postnatally, right? So slightly under 1 × 10⁻⁴ vector genomes per. And it can still be on a weight basis, right? You can estimate the weight of the fetal recipient, whether it be in a mouse model or a large animal model. But you need to be very in tune with the fact that toxicities may be better or worse in the fetus, and and that's the whole point of the preclinical studies that need to be done, and that continue to need to be done before we contemplate moving things forward.
I just have one comment to add, if I may. For early postnatal development, when there are clinical trials in the young patient population, in pediatric patients, you need to ensure that there's the prospect of direct benefit I'm fairly confident that that would apply to fetuses as well. So the dose issue, although it may be difficult to do interspecies comparisons, I think it is an important issue that one would want to select a dose that has the chance to show efficacy based on whatever data that are available.
And then I have an additional question here for Chris. You demonstrated that we should be checking for germline integration and show that for particular vectors, germline integration is a very rare event. Presumably, though, this needs to be addressed for every single type of delivery vector or vehicle that we're thinking about. What do you think are some of the key experiments that are needed to be done as we're exploring these novel therapeutic approaches?
It's a difficult question. So I think not being, I guess, a reproductive biologist, I'm not sure exactly what animal model would be the closest replicate of a human, but I would guess we would want to pick an animal model that most closely recapitulated human reproductive development in order to look at the risk of germline modification, especially since it seems, depending on which species we're using and which vector we're using, we may get completely different results. Like Dr. Tarantal and I got completely opposite results using monkeys versus sheep and using MMLV versus lentiviral vectors. So I would think the first thing, certainly we would want to make sure we're using the vector that's going to be used clinically to do the test to look to see whether the germline is modified, and try to do that in an animal model that we think is the most predictive of the human. And I'm not really sure which animal that would be that we could say with certainty, because we saw an incidence of 1 in 10,000 or something in this animal, that suggests that that's what we're going to see in humans. I know Grasa and I at one point had discussed using tissue organoids, because at Bufirm we actually have people that work in fertility that have developed testes organoids and ovarian organoids, and perhaps use that as a way, because that would actually be a human system with human cells that we could look at what the risk is, or what factors determine whether the germline would be modified by a specific vector to be tested in the clinic. But otherwise, I'm not sure what else to say about that. I mean, it's a very important question, but I'm not sure what the definitive study would be that would tell us, okay, this is safe enough that we can move forward.
And it seems it's a little unique to each of the delivery modalities, or to each disease even, given that some have just different risks than others. And so paying attention to where we predict the highest risks may be. And then a corollary to that is, what do you think we should be monitoring for in patients once we are developing these clinical trials? What are some of the assays that we should be making sure to include as we look for safety in addition to efficacy? And how long should we be monitoring patients for? Any thoughts?
Well, I think for the first patients, we'll need to monitor them for a long time, right? And depending on the technology that we are using, the way of monitoring these patients is going to be different. Of course, if it's hematopoietic stem cells, those are the easiest ways, or one of the easiest ways to monitor, because you can take blood and you can analyze the cells for anything that may happen. If we are delivering the therapy to the brain, or to delivery, may be more difficult to follow safety unless, I mean, through ultrasound, through biomarkers would be the only way to follow these patients. But production of protein, I mean, the same safety parameters that we use for adults probably would be used for the same after birth for those patients, to follow them and for maybe a longer period of time to ensure that those patients, that the therapy is safe. But I think what we do right now after the patient is born, what we do right now for children and adults, it would be a good way to monitor these patients. I don't think, unless any of you think about any extra monitoring for patients that would be transplanted in utero versus, or treated in utero versus postnatal.
I agree. Oh, so sorry, go ahead, Chris.
Oh, I was just going to say one thing. I guess it's not unique to in utero, but I know an issue that's come up with all of the clinical trials with AAV for hemophilia is that we really don't have any idea what the rate of genomic integration is in humans, because there isn't any way you can sample the liver of a patient with hemophilia, even doing a small biopsy or something to look at that. And like the recent study by Denise Sabatino looking at the dogs over the course of 10 years, she mapped something like 1,700 AAV integrations in the liver of 4 of those dogs. And many of them actually fell within regions of the genome that have been linked to human cancers, but none of the dogs developed an overt tumor. So if she hadn't euthanized the dogs and taken out the liver to do the DNA analysis, she never would have known any of those integrations happened. So that, I mean, I don't have an answer to that, but I think that's a really important safety issue that I'm not really sure how we can address. I mean, we have all these patients that have been dosed with AAV and we have no idea whether it integrated at all in the liver or what incidence or where it's integrated unless they develop some obvious sign of pathology that leads the physician to go in and look at the liver.
Yeah, I was going to actually say something similar. I think 2 points. One, we're just going to learn a lot from postnatal gene therapy trials, which are still in their infancy. So it makes it a little bit difficult to answer the question, right? And so, you know, there's a reason why, as was alluded to earlier, risks and safety concerns about gene therapy in general only get amplified when you're talking about doing that before birth, right? And so there's a reason why postnatal gene therapy trials go first, and we learn a lot from them. And so how long and what we monitor we will learn from results of those trials, I think is the one important thing. And then, like Chris alluded to, there's increasing awareness of AAV integration following in vivo delivery, right? And then there's some biologic rationale to think that that integration may be more significant if you do it earlier in life when things are proliferating. And then the corollary to that is the concern about integration is integration into hotspots that may predispose to oncologic risk, right? And so that's what Denise's study showed, even though, like Chris said, they didn't have tumors, there seemed to be clonality of the cells where there was integration in these hotspots. And so if then we think about applying gene therapy in the early neonatal period or prenatal period, I think it just speaks to the need for lifelong monitoring if possible, right? That's the ideal situation.
Yeah, and I think an important part of this is just consent. Everything we do or don't do has risks and benefits to it, and making sure that we have very clear conversations with patients and families as they think about treatments or not doing treatments, because there is just always some question of things that we don't know and that we will or may learn along the way. And I think this group has done a really nice job thinking about that, and really engaging many different aspects of the community to make sure that we're properly informing and educating patients and involving many different types of care providers in that process.
Yeah, exactly. And that's where the key point of disease selection comes in, right? And so, the very first study that we did here with Kiran Musunuru for gene editing looked at targeting PCSK9 for hypercholesterol. There's no way we would ever think about doing that into a fetus. So it's just a nice proof of concept study, right? It also helped establish a great friendship and a nice collaboration, which was an added benefit. But to your point, and what has been mentioned throughout the morning so far, it's a matter of weighing risks and benefits. And for diseases where the outcome is terrible, then maybe you accept a little bit more risk, as the patient families alluded to early at the beginning of this conference, right? And so I think it's a key is disease selection when we're thinking about things for the future.
And how do you guys think about number of patients to treat, given that potentially there could be benefit to many patients versus waiting to see what the outcomes are for smaller numbers initially?
I think the FDA they provide some guidance as to how many patients you should enroll in your initial phase 1 to look at safety and potentially efficacy, don't they? I mean, I was thinking of the pre-IND that Grasa and I have submitted for hemophilia. I think we were targeting 10 patients, weren't we, Grasa? Or
I mean, but for some of these more rare disorders, right? It may be difficult
to find 10 people.
depends on the prevalence of the disease as well. But I'm sure that, I mean, we get that guidance from the FDA to tell us how many patients we will need to enroll.
Something that I'd like some guidance with is, what bar of safety do you have to demonstrate to move on to clinical translation? So as an example, separate from genetic diseases, if you have a 3-year-old with leukemia, do you give them chemotherapy or let them die? We give them chemotherapy. And you now have a 95% survival for a lot of these. You are committing germline graffiti with that therapy. You predispose them to getting cancer. You predispose them to having heart failure. And so to say, are we creating a totally different logic for these sets of diseases? Or do we have to hold ourselves to the baseline risk of, germline mutations? And clearly I don't have the answer. I'm just posing the question. But do you need to see no off-target effects? Do you need to see 1 in 50 like Chris was showing in sperm? Do you need what's the bar of safety when you're up against lethal genetic diseases?
I think it's a great point, Dave, but you have to also think about alternatives, right? So the kid with leukemia maybe doesn't have an alternative for a treatment, as you graphically laid out. But I'm the last person that wants to be unenthusiastic about in utero gene therapy or gene editing, but for a lot of, and this is maybe the point that was laid out earlier, for a lot of these diseases, there's a potential for testing a therapy postnatally to demonstrate its safety, and then moving to the prenatal stuff. So I think it depends on the alternatives that are available.
Yeah, I agree with you. I think that's in the equation, but I am curious to know, what level of safety we hold ourselves to.
I can't address some of the toxicities that one might see specifically with gene therapies, but in the antisense world and for drugs, we are certainly aware that the potential benefit is something that needs to be kept in mind when you're talking about risk. The kinds of toxicities that we see that either limit the dose that can be administered to humans or just administering the drug at all are things that are severe, serious, like death, or unmonitorable, like such severe, like seizures, convulsions, such unacceptable CNS toxicity that animals have to be sacrificed prematurely. Those are the kinds of things that would that usually will limit or halt a clinical trial, in my experience. But short of that, especially if there's a way to monitor carefully, that's done very carefully and does not typically limit the trial.
There was a question that was asked about how are these conditions screened, diagnosed, and identified. I think there were some nice sessions earlier that talked about that to some degree. But I'm wondering if similarly during the period after the therapies are delivered prenatally, what kind of monitoring do we do to the rest of the pregnancy versus after the fetus is born? Are there specific things that we should be monitoring during the remainder of the in utero period? And are there ways to do that apart from kind of standard pregnancy monitoring?
I'm not aware of any type of monitoring that depends on the disease, right? And you monitor the patient for that disease if he's still in utero. So I think still the clinical, what is in place for the clinical protocol for that disease during gestation, I think still applies for gene therapy or any other fetal disease.
Mm-hmm.
I would agree. I think it's a standard imaging technology. I would argue that we wouldn't want to sample anything from the fetus after the therapeutic was delivered to see how well it's working. I mean, unfortunately, there's going to be a period of just making sure that the mom and the fetus are doing well clinically, and we'll see if the therapy works after the baby is born. I don't think you subject the mom or the fetus to any increased risk for diagnostic purposes via an intervention after the therapy is delivered.
Any last thoughts that anyone has or comments that they want to share as we're running out of time?
Actually, I just had one quick question, I guess. Bill raised a really interesting point that hadn't occurred to me. So there actually isn't any animal model that we could use to look at off-target effects of gene editing, right? Because the genome sequence is different in every species. So even if you had something that worked perfectly in non-human primates, there's still a possibility that it would have an off-target effect in humans. I mean, do you have any suggestion how we would look at that? We would have to do just in silico prediction for whether it would have similar specificity in human, or? I didn't mean to put you on the spot. I just thought it was a really interesting point that you raised because it hadn't occurred to me.
Yeah.
Yeah.
So I'm sure Theodore, who's part of a panel later on in the day, probably has some very knowledgeable insight here. But basically, I agree with you. There's no animal that replicates the human in every cell and every organ. There are humanized liver mouse models. So you could, I guess, look at off-targeting of hepatocytes in a humanized mouse model in vivo. I think a lot of the off-targeting is done ex vivo to look at specific human off-target sites. I'm looking forward to hearing Theodore speak later on today and his insights.
Well, thank you so much all for joining today, for your wonderful presentations and for this panel discussion. We're looking forward to the rest of the presentations and for continued conversations. So thank you, everyone.